Okay, so as I kind of mentioned a little bit and hinted at, there's a lot of complexity to the structure of proteins. So, in order to describe that complexity of the structure, we break it down into levels. So, uh, we describe proteins in terms of levels. But first off, let's describe what a protein is. I keep saying peptide. So, did you notice I said tripeptide, tetrapeptide, everything like that? Peptides are just small proteins. So, proteins are specifically a polypeptide. So, peptide is like something that when you just have a couple amino acids, you would call it a peptide chain or a peptide. A protein is a polypeptide with 50 or more, 50 plus amino acids. And it typically, or in order to call it a protein, it has to have biological activity. So if you have a really, really massive protein, or I'm sorry, really massive peptide, then you call it a protein. Okay, so a primary structure is what we talked about just a moment ago, where you make peptide bonds. So the purpose of a primary structure is to show the sequence of amino acids held together by peptide bonds. So just above, we were talking about this sequence of amino acids was a tyrosine, serine, valine, asparagine. That is the sequence in the order that you see those amino acids show up. So the primary structure is the sequence of amino acids held together by peptide bonds. So what this is important for is biological activity depends on the primary structure. So for example, glutamine, histidine, proline. This particular amino acid in this order is a peptide hormone, and it stimulates the release of thrioxine. So thrioxine is a hormone in produced and released from your thyroid, and it helps in regulation of your metabolism. It helps in your brain activity. There's a lot of different purposes for thrioxine. And this particular peptide is what signals your brain to make more or release more of that hormone. However, if you think about it, so glutamine, histidine, and proline, you could have a lot of different orders of that. You could put proline first, then histidine, then glutamine. You could switch them all around. So there's 6 possible orders for this peptide. With glutamine, histidine, and proline, one of each of them. However, only glutamine, histidine, proline has activity. So these are very specific. The primary structure dictates its reactivity in the body. And then additionally, some history lessons for you guys. In 1953, the first primary sequence of a peptide was determined. It was the first time we could actually tell what these things were, and it was insulin. So insulin is a hormone in your body that regulates your blood glucose levels. So people who are diabetic will typically take insulin in order to help regulate their blood sugar levels. This is completely off topic, but my friend in the lab, he used to be one of my coworkers. For some reason, I can smell insulin. It has a particular smell to me, but he couldn't smell it at all. And I could always tell right after he had lunch because I could smell the insulin. That's a little weird, but that's okay. So this is just a picture form of primary structure for you guys. So this particular peptide chain or protein overall has just over 50 amino acids. It has 2 chains of peptides. So you can see one's in blue, one's in purple. So let's do blue for chain A. So chain A has 21 amino acids, as it says here. And you can see the very specific orientation of them. You see that here it shows, here it says H2N or H3N, I think it says, it's hard to read. So it says H3N, showing that this is the N terminus, and then you start with a glycine, isoleucine, and this shows the exact order of all of the amino acids in this peptide chain. And then you have a second chain of amino acids, so we'll call it chain B. It has 30 amino acids. And again, you can see you have your N terminus, your C terminus, and the exact order of those peptides. And then additionally, let's do orange. You can see that we have cysteines in here. And where these cysteines are, we have a particular, that's just how we're combining the two chains together, is through covalent bonds, through these disulfide bonds. So wherever cysteine is, you have that free SH, or that thiol on the end of that amino acid. It can then do oxidation and form disulfide bonds. So if you remember way back when, this is why we learned this reaction. If you take a thiol and you oxidize it, you form a disulfide bond. It's the exact same process. This is why we learned it before without all of the fancy junk around it. It's the same exact reaction. The thiol, two thiols oxidize together to form a disulfide bond.